You want to hear me? All right. Good morning, everyone. Thanks, Morgan, for that great talk. Apologies to those of you who were at my evolution talk. Some of this may be a little bit familiar to you. So I'm going to be giving an update on some of the results that we've gotten thanks to some of the genome resequencing and then obviously the Manacus vitellinus genome assembly. So I don't think I have to convince anyone here of how awesome sexual traits can be. You know, amazing colors and plumages and behaviors and structures. And despite how much interest there's been over the years in sexual traits, we really don't know all that much about the genetic basis of a lot of these traits. And because mannequins are so cool, and because there are certain population patterns that can help us uncover the genetic basis of these traits, I think mannequins are a great study system to use to try to do some of this. So the Manacus genus is a little odd, green thing, but so you may be familiar with this genus, but just going sort of clockwise, starting on the right, Manacus, that's the white-bearded Manacus, the orange-collared, candel, the white-collared, and then vitellinus is the golden-collared. And they can be thought of sort of as a super species, right? So they replace each other geographically, and everywhere they come into contact, they form a stable hybrid zone. White-bearded Manakin, some of us heard them this morning. I don't know, maybe the people who went out looking for Manakin saw some. And so I'll show you, oh no. Well, you can watch this video, this kind of trippy colored white collar Manakin. There's a female watching. And they have this acrobatic display where they jump back and forth, snapping their wings and potentially cockroaches. Over this display court, which is weird ground on the forest floor, the male weird between these cattle, and there's the snapping. Okay, so just to zoom in on that range map that I showed you. Well, so first, let me say there's really strong unequal mating success among males. So there's the potential for really strong sexual selection. So zooming on that range map where white-collared, golden-collared Manakin ranges come into contact in western Panama, if you were going through the forest heading west, you'll find golden-collared Manakins going through these numbered populations that Mike and his students have studied over the years. Golden-collared, 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 and then you'll go across the Rio Changanola and find white-collared Manakins. And so you'll say to yourself, well, here's the species boundary, right? And these measures of plumage color will agree that these transitions in both the collar, the yellow to white, and the belly from olive to yellow occurs right at that dashed line there between populations 3 and 4, which represents the river. However, in the 90s, it was discovered that the actual genetic transition between these species occurs between populations 8 and 9, which is represented by that dashed line. And so you actually have a region in between the hybrid zone and the river where birds look like golden-collared Manakins, but they have a white-collared genetic background. And so this is a great sort of opportunity to study a lot of things, but in particular to study the genetic basis of these plumage traits, because you've got populations right on either side of the river to be very similar but different in their plumage traits. So this is the question that we had going in: what gene or genes determine this plumage polymorphism? And this is data that was originally collected by H.C. Lim, who some of you know, the former postdoc in Mike's lab now at George Mason University, did this sequencing. And then I've been analyzing this data. So just to give you a lay of the land here of these populations. So population 2, we're considering sort of the parental white-collared population. Population 12 over there in Gamboa, sort of the parental golden-collared. And then populations 3 and 4 right on either side of the river, we have 10 individuals from each of those populations. And they're both, the genetic background is candii, one has a golden collar and one has a white collar. So then I've been doing these analyses looking, well, so let me also say the sequencing, this is whole genome resequencing about 15x and because there's reference assembly, we can map to the assembly, and then find SNPs and indels. So I've calculated divergence between the populations, nucleotide diversity, and then we can look for regions that might contain genes of interest. So here's the FST plot. So just looking across the genome, alternating colors are different scaffolds from the reference assembly, and each dot is a 10 kb window. And so you can see that there are sort of two really tall peaks here and here, and then a few other sort of medium height peaks. And this is between populations 3 and 4. This is this FST. So these two populations right on the other side of it. And it turns out that a lot of these medium height peaks are actually on the Z chromosome. So as we talked about a little bit, this is the sex chromosome, right? So there's an expectation that divergence may happen faster on the Z because it has a smaller effective population size, but there may also be other interesting things going on with Z species that have really strong sexual selection, like manakins, because males have a smaller effective population size. But I won't go into that anymore. It's just sort of an interesting thing that we can look into a little bit more. I'm just going to focus on these two main tallest peaks here. One of them is on chromosome 2, one of them is on chromosome 24. And in the interest of time, I'm sort of going to focus on this one, the tallest one, that is a region on chromosome 24. And I'll just zoom in on that. So the top panel is FST, that's relative differentiation. It takes into account allele frequencies in both populations. DXY is sort of absolute differentiation, just specific, literally how different are the genomes in these areas. And you can see they both have this really narrow peak, really tall peak right in the same spot, about 6 megabases along the scaffold. The scaffold is about the size of the chromosome, so about 6 megabases along the chromosome. So we know that these two populations are really different right at this area. Another thing that we know is looking at nucleotide diversity, that population 3 kind of has a normal sort of, doesn't have any obvious dips in nucleotide diversity except right at the ends of the chromosome. Population 4 that has this golden collar looks like it might have a sort of a dip in nucleotide diversity right at that same spot. And if you just divide one by the other, you can see that there's this sort of spike. Looking at population 3 divided by 4 nucleotide diversity, there's a spike right there at 6 megabases. And so what we can gain from looking at nucleotide diversity is if there's been a selective sweep, you would expect to see a reduction in nucleotide diversity at that region. So this is a little bit of evidence that there could have been a selective sweep in population 4. So this is another thing that we might expect to find if there are genes underlying this in that population. And then last, we can look and see, so we know that they're different in this region, but are they different in the way that we would expect? So are the golden collared birds similar to vitellinus? Are the white collared birds similar to parental candel? And so looking across this region, you can't really read the numbers, but this is about 1.2 megabases across that region. Population 2 and 12 on either side are the parental white collared and golden collared individuals. Each row is an individual, and each SNP is coded with a color. So this is just looking at sites where parental golden collared and white collared populations are fixed different from one another, which is another thing that we would expect to find in sites that are associated with the collar type, right? And you can see that looking across, there's this sort of narrow region where these population 4 birds are very similar to vitellinus. Population 3 birds, with the exception of 2 heterozygotes, are very similar to candel. So we know in this narrow region right around 6 megabases along the chromosome, the two populations are very different from one another. They're different in the pattern that we would expect in terms of which population is similar to vitellinus, which is similar to candii. And we know that population 4, which has the golden collar, looks like it has some signatures of a selective sweep. So now we can look at the genes and see if there's anything we're looking at. These little bars at the top are exons of genes. And right in the middle of that region is this gene called BCO2. You may recognize the name because Moni showed us some statistics about it in crypto last night as an example. So it's called beta-carotene oxygenase 2, and what it does is it takes in carotenoids and it chops them up into colorless products. And it's actually been associated with white-yellow polymorphisms a number of times in the past. So the food industry has given us some examples. In sheep, a mutation in BCO2 can turn that fat from white to yellow. This is cow milk fat. In cows without a functioning BCO2, carotenoids build up in the milk fat and it turns more yellow. Can't quite see it on this screen, but and then in rabbits as well, commercial rabbit farming, mutations in BCO2 result in yellow fat rather than white fat. But we also have examples from other organisms. So as close as you can get to a wild red jungle fowl will have gray skin, whereas domestic chickens have yellow skin. And this difference has been mapped to a regulatory region just upstream of BCO2. So, and then last but not least, in this paper showing that golden winged and blue-winged warblers, which are very, very similar across the genome and hybridize extensively, are only different at 6 points in their genome. And one of those is over the same gene, BCO2. So I think even though we don't have the same kind of good candidate genes like MC1R for melanism that we do for carotenoids, we can start thinking about BCO2 as sort of the MC1R carotenoid coloration. In the interest of time, I'm not going to go over that second peak because it looks like we're almost out of time. But I would love to talk to people about this one later because it's also really interesting. But so I'll just give you some takeaways. Manakin genomes can give us really cool information about a lot of different things, but including the evolution of color traits. And where we're going with this next is we want to look and see, is the vitellinus trait independently derived in vitellinus, or is it potentially the result of introgression from Aurantiacus, or is it potentially Aurantiacus's golden collar the result of introgression from vitellinus? And then we're also going to look into the female preference for golden collars, and I would love to talk to some of you more about that later on during the conference. All right, thanks everyone. Thanks for your attention.
What do you know about the regulatory elements